Hi students, I'm going to go through the instructions for how to complete the Unigriffin assignment. Please open your notebook to your amino acid decoding chart. You can see here the page. If you don't have this handout, go ahead and go over to the extra handouts box and see if you can find one in there. Okay, next step is to go to Google Classroom. And in Google Classroom, you're going to see that in the assignment, I've given you the drawing template. Now that you don't need to open because you have a paper copy available to you in the classroom. We have the Unigriffin Instruction page where you found the video you're watching now. And then also we have the Protein Synthesis Google Sheets. That's what you need to open. That's going to be where your assignment is today. When you open it up, it's going to look like this. A unigriffin is a mythical creature that is a hybrid, meaning that it has genes from multiple different animals. It has genes from birds, lions, and unicorns. So scientists have recently obtained samples and have mapped out 10 of the genes. Your job is to analyze the DNA sequence of the unigriffin samples in order to determine which features each sample codes for. You're going to determine which traits each type of unigriffin has by decoding the DNA. There are 10 possible genes. You can see them here in this table. Okay, so the 10 genes are listed out. There's a gene for ear shape, wings, front legs, beak, body color, eye pupil, unicorn horn, body feathers, and tail. Each of those genes has 2 possible variations, which produces 2 possible amino acid sequences during protein synthesis. For example, ear shape could produce an amino acid sequence of valine, serine, leucine, or valine, serine, lysine. Now, depending on which protein is made through protein synthesis, that will determine which version of the trait your unigriffin has. For example, if it's valine, serine, leucine, your unigriffin will have rounded ears like a lion, whereas if you go through the process of protein synthesis and translate the DNA into valine, serine, and lysine, then your unigriffin will have pointed ears like a horse. So you will be using your knowledge of transcription and translation to determine what the amino acid sequence is and therefore what kind of traits that unigriffin must have. Okay, so for this next step, we need to look down here at the bottom. There are 2 tabs here. You see one says Intro, Traits, and Drawing. That's the one you're on right now. We need to click on the one that says Transcribe and Translate. When you do, you'll be brought to this page. Now notice at the top, there are instructions. So if you get stuck, you could always read those, or you could pause this video and rewatch the parts that you need to watch in order to go through step by step. Okay. In the upper left here, you're going to see an amino acid chart. You're welcome to use that one or the one in your notebook. They have the same information. It's just whichever one you're more comfortable with. Okay, so looking here, we have unigriffin 1, 2, and 3. Guess what? You're just going to transcribe and translate the DNA code for one of these 3 unigriffins. So coordinate at your table so that you're not all doing the same one. Okay, someone should be doing 1, someone should be doing 2, someone should be doing 3. And if there's 4 people at your group, then someone will double up, and that's okay. Alrighty. So for each of these, you can see here that the DNA sequence is given to you, and it's broken into codons or sets of 3 nucleotides. Your job is to transcribe the DNA into mRNA and then translate the mRNA into the amino acid to then figure out what kind of trait your unigriffin will have. I'm going to use one example. We're going to go through one of the genes together, okay? So for DNA, if we have TAC and we want to transcribe it, T bonds with A bonds with U, C bonds with G. C bonds with G, A bonds with U, G bonds with C. T bonds with A, C bonds with G, G bonds with C. T bonds with A bonds with U, T bonds with A. Okay, now what if I had done something wrong? Let's say that I had said that A bonds with T. Let's look at what happens. It's going to turn red. So if you get a box that is red, that's telling you you did something wrong. And I want you to go back and refer to your amino acid chart and your codon base pair rules to see what you did wrong and then correct your error. Okay. I recommend that you go through and you transcribe the entire DNA sequence all at once, so all the way down to the bottom, okay? I'm going to stop here and just show you what the next step is. So here, now that we have our mRNA, we can now use our mRNA amino acid chart to determine which amino acid will be put in that place. So you can look at the chart again here on the screen or the one in your notebook, but remember that you start in the middle, so A is in the middle, and then it goes to U, and then it goes to G, and in this case, it says methionine or start. So in this program, you just type in the three-letter abbreviation, don't type in the whole name, okay? So met means start. Every gene is going to start with a methionine, and up here it says this at the top, okay? So now I'm going to look to GUC. So GUC says valine. Now AGC is serine. AAA, we have AAA is lysine. And then UAA says stop. Okay, now if I type in stop, it's going to turn red. So look up here. It says when you reach a stop codon for a gene, there will be no amino acid. You're going to write STP. Okay, so let's see what happens if I change it to STP. It's not red anymore. Okay, I know that I've reached the end of my gene because I get the stop code. So everything between the methionine, which is the start, and the stop, that's going to be the protein. So in this case, this protein is 3 amino acids long. It's valine, serine, lysine. Okay, so what I want you to do next is I want you to compare this amino acid sequence to the information that you get here. So on this handout that you can get from the front of the room, on the back page, there is this chart of all the different genes. It's the same information that you have here. Okay, I just put it on the paper to try to make it a little bit easier for you. Okay, so what I want you to do is I want you to compare the amino acid sequence, and I'll put this side by side so you can see it better. Okay, so for this amino acid sequence, we had valine, serine, lysine. So that's gene 1. Let's see. Do I have valine, serine, leucine? Nope, I have valine, serine, lysine. So that means my Unigriffin must have pointed ears like a horse. I want you to either highlight that or make a check mark next to it somehow to keep track that that's the version of the trait or the phenotype that your particular Unigriffin will have. Because your last step after you figure out for the rest of the 10 genes by completing the transcription and translation in your code here, the last step is going to be to draw your Unigriffin. Okay, so you're going to use all of those traits that you've identified for the Unigriffin here, okay? Like, for example, pointed ears like a horse, or maybe you have short wings or long wings. Either you have scaled front legs or furry front legs, okay? So depending on what your gene's coded for, you're going to then draw what your Unigriffin will look like. And then here, you're going to pick one of these examples, okay? So here's our traits, okay? So for example, I'm going to show you my example here, okay? So you could say, okay, there we go, okay. So my example here was eye pupil, okay? So you can pick any of these, but I picked eye pupil for this example, okay? So for eye pupil, I had the phenotype round pupils, okay? So my amino acid sequence, I got it right from here, okay? So it's here it is right here, and I've just written it in on my paper, okay? And then I looked up on my code here for that gene, and I typed in, or I wrote in the exact DNA sequence that coded for that trait, okay? All right, so last step is you're going to turn this into the tray in the front of the room. And then once you do that, I want you to check and make sure you completed your assignment from last class, which was number 59, modeling protein synthesis, and also that you've completed the Ed puzzle on protein synthesis. And that's it for today. Thanks so much.